Welcome to our lecture on the diversity of chordates. Today, we are going to explore a group of animals with a life story that sounds like a science fiction movie. Imagine being born as a highly active, swimming creature with a complex nervous system, only to grow up, glue your head to a rock, absorb your own brain, and spend the rest of your life as a stationary bag. These bizarre and fascinating creatures are called Urochordates, and they hold the key to understanding our own evolutionary history. Now that we have introduced these bizarre animals, let us look at what they actually are. Urochordates are commonly known as sea squirts or tunicates. Even though they look like simple sea sponges sitting on the ocean floor, they are actually our distant evolutionary cousins. They sit right at the boundary between simple, spineless invertebrates and complex animals with backbones. By studying them, scientists get a unique window into how the very first chordates, the broad group that includes humans, might have evolved millions of years ago. Understanding their evolutionary position brings us to their formal biological classification. Urochordata is a subphylum within the phylum Chordata. The name itself is a massive clue about their anatomy. It comes from the Greek words ora, meaning tail, and chorda, meaning cord. This perfectly describes their defining feature. They possess a stiff supporting rod called a notochord that is strictly confined to their tail region. However, this advanced chordate feature is only present during their early free-swimming larval stage, which sets the stage for a dramatic life change. This temporary larval complexity leads us to one of the most dramatic transformations in the animal kingdom. As these active, fish-like larvae grow up, they undergo a process called retrogressive metamorphosis. The word retrogressive means moving backward. Instead of developing more advanced features, they actually lose them. They completely absorb their notochord, their dorsal nerve cord, and their postanal tail. They attach themselves to a rock and transform into a simple, stationary organism whose sole purpose is to filter water. It is a perfect example of evolution favoring simplicity over complexity when it suits survival. After this dramatic backward transformation, what does the adult actually look like? The adult body becomes enclosed in a tough, leathery outer covering called a tunic, which is why they are often called tunicates. Fascinatingly, this tunic is made of a substance called tunicin, which is remarkably similar to plant cellulose, a very rare feature in animals. Inside this protective bag, they possess a specialized feeding structure called a branchial basket. This basket is lined with numerous gill slits, acting as a giant sieve to continuously filter microscopic plankton from the surrounding seawater. Knowing how they feed naturally raises the question, why do we care about these stationary bags on the ocean floor? In the real world, urochordates are vital to marine ecosystems. Because of their branchial baskets, they act as highly efficient biological water filters, cleaning the ocean by consuming vast amounts of suspended particles and phytoplankton. Beyond ecology, they are incredibly important to human medicine. Some tunicates produce unique chemical compounds that scientists are currently researching and using to develop powerful anti-cancer drugs. So these simple sea squirts are actually saving human lives. To truly appreciate their ecological impact, we need to quantify how efficiently they filter ocean water. Marine biologists measure this using a mathematical concept called the clearance rate. The formula is CR equals V divided by T multiplied by the natural logarithm of C0 over CT. Conceptually, this calculates the volume of water completely cleared of particles per unit of time. CR is the clearance rate in liters per hour. V is the total volume of water, and T is the time elapsed. C0 is the initial concentration of food particles, and CT is the final concentration. The natural logarithm accounts for the fact that as the C-squirt filters water, food becomes scarcer, making it exponentially harder to find particles over time. If the C-squirt grows larger, the clearance rate increases. Before we move on, let us see this formula in action with a concrete example. A marine biologist is studying a sea squirt in a 10-liter aquarium. Over a 2-hour period, the phytoplankton concentration drops from 500 cells per milliliter down to 100 cells per milliliter. We need to find the clearance rate. First, we substitute our given values into the formula. We replace V with 10 and T with 2. Inside the natural logarithm, we place the initial concentration of 500 over the final concentration of 100. Dividing 10 by 2 gives us 5, and 500 divided by 100 also simplifies to 5. Using a calculator, the natural logarithm of 5 is approximately 1.609. Multiplying this by 5 gives our final answer. This single sea squirt clears over 8 liters of water every hour, proving how ecologically vital they are. Now that we have calculated their feeding efficiency as adults, let us look back at the dramatic transformation that got them there. During retrogressive metamorphosis, the Urochordate larva literally destroys and absorbs its own tail, notochord, and nerve cord. Because these structures make up a huge portion of the larval body, this process results in a massive reduction in their overall physical length. To truly grasp the sheer scale of this biological recycling, biologists calculate the percentage of body length lost when the free-swimming larva shrinks down into a juvenile sea squirt. To quantify this extreme physical change, we use a standard mathematical formula for percentage decrease. The formula is P equals Li minus Lf, all divided by Li, and then multiplied by 100. Conceptually, the numerator represents the absolute physical length of the tail and tissues that were absorbed by the animal. Li stands for the initial length of the active larva, usually measured in millimeters. Lf represents the final length of the newly settled juvenile. By dividing the absolute difference by the initial length, we scale the loss relative to the starting size. Finally, multiplying by 100 simply shifts the decimal point to give us a recognizable percentage format. If the final length is very small, the percentage loss will be very high. Let us apply this formula to a specific biological specimen. Suppose a free-swimming larva has an initial length of 4.5 millimeters. After settling on a rock, the new juvenile sea squirt has a final length of just 1.2 millimeters. We want to find the percentage of body length lost. First, we substitute our given measurements into the formula. Subtracting 1.2 from 4.5 shows that the larva absorbed exactly 3.3 millimeters of its own body. This was mostly the muscular tail and notochord. Next, we divide this 3.3 by the initial 4.5, giving us a decimal of 0.7333. Multiplying by 100 gives our final answer. The animal sacrifices over 73% of its original body length, physically illustrating just how drastic this retrogressive transformation is. Now that we have calculated the massive physical loss, let us look at exactly what this transformation looks like visually. On the left side of this diagram, you see the active larva. Notice the long tail containing the notochord and the dorsal nerve cord. These are the advanced features that link them to vertebrates. Following the metamorphosis arrow to the right, you see the adult form. The tail is completely gone. The animal is now glued to a surface. You can clearly see the tough outer tunic protecting the body. Water enters through the top incurrent siphon, passes through that large mesh-like bronchial basket where food is filtered out, and exits through the side excurrent siphon. This diagram perfectly captures the sacrifice of mobility for highly efficient filter feeding. We have covered a lot of ground today, so let us summarize the most important ideas. First, Urochordates serve as a crucial evolutionary puzzle piece, showing how complex chordate features can exist early in life and be lost later. Second, their defining characteristic is having a notochord strictly in the larval tail, which they completely absorb during retrogressive metamorphosis. Finally, while they may look simple as adults, their branchial baskets make them incredibly efficient biological filters that keep our oceans clean and balanced. You now have a solid understanding of the life cycle, anatomy, and ecological importance of the Urochordata. Great job following along with both the biology and the mathematics today.